Welcome back to the deep dive. Today we are taking you inside a mystery that was solved by, well, 2 full decades of just unyielding dedication. It's a discovery that really promises to reshape agricultural practices around the globe.
It does, and this deep dive is centered on what we've been calling the hunt for the glowing assassin.
The glowing assassin, it sounds dramatic.
It sounds dramatic, but you know what? It's entirely accurate. We're talking about a microscopic foe that took 20 years of persistence not just to find, but to actually commercialize.
And the mission was to find a definitive killer for one of agriculture's most relentless enemies, the spider mite. And the solution, I mean, it wasn't found in a lab test tube. It wasn't scraped from rich topsoil, which is where you find most of these microbial agents.
Right, the usual suspect.
Exactly. It was discovered hiding in one of the smallest, most, inaccessible places imaginable.
In the throat of an even smaller parasitic worm.
An incredible...
It really is. This is the revolutionary story of the microbial agent Photohabdus luminescens, the light rod bacterium. And this deep dive, it's really centered around the incredible work of Taiwanese researcher Dr. Shifeng Jia and his team. They're the ones who successfully isolated and, crucially, stabilized the effective components.
Creating the world's first commercially successful microbial acaricide from this very specific organism.
The very first.
Okay, so let's unpack this. What the research is telling us is that the 0805 P2R strain of this bacterium, it kills mites in a way that is it's utterly devastating to the pest.
Oh, absolutely. The description is that it causes their gut tissue to rupture. They're described as being, and this is a quote, intestinally punctured and rotten.
Wow. And yet, despite this, incredibly violent action on the mite, it's completely harmless to us.
Completely. Harmless to all mammals, and this is critical for farmers, to the beneficial insects they actually rely on.
That's the core paradox, isn't it? And the genius of the whole breakthrough.
It is. So our mission today is to really get a comprehensive understanding of 3 key things. First, just the sheer painstaking difficulty of finding and isolating this glowing organism.
Right.
Second, how its unique, sort of natural Trojan horse mechanism actually works at a complex molecular level to destroy the pest.
And finally,
why this 2-decade commitment is such a monumental, globally significant step forward for sustainable pest management. We'll be bringing in some key context from other recent agricultural research that really just illustrates how urgent this solution is.
Okay, so before we celebrate the assassin, we have to really appreciate the target. Why is the spider mite such a huge, intractable problem? It's tiny, yet it seems to defeat millions of dollars of chemical research every single year.
Well, the target pests are typically leaf mites. Most commonly, you're looking at red spiders, Tetranychus canzae, and the 2 spotted mites, which some people call white spiders.
And these are just universally hated.
Right. They're universally recognized by the entire agricultural community as the most difficult pests to remove. And that's for sophisticated biological reasons, not just because there are a lot of them.
So what's the mite's secret weapon in this whole evolutionary arms race?
It really comes down to their speed. Their speed and their reproductive cycle, which allows for just extremely rapid evolution.
Okay.
Mites have incredibly strong reproductive capabilities. They lay hundreds of eggs, and they combine that with very short generation cycles. I mean, in ideal conditions, a generation might cycle through in less than a week.
Less than a week. So when you introduce a chemical, which is often targeting a very specific biochemical pathway, you're immediately applying this intense selection pressure.
Precisely. If even a tiny, tiny percentage of that population has a random mutation that gives it some slight resistance to that chemical, they survive.
And then they take over.
They take over. Because the generation cycle is so short, those resistant individuals can reproduce so fast that within just a few seasons, sometimes even just a few weeks, they establish a population that is entirely immune to that original chemical.
It sounds like just throwing money down a drain. You buy a new chemical, it works great for a few months, and then you're right back where you started.
That is the reality. Our sources highlight that in Taiwan alone, there are 19 approved chemical miticides in circulation. 19. Yet they frequently fail to provide effective long-term control. And this failure, it forces farmers into this really detrimental rotation pattern, constantly switching between chemicals to try and stay ahead of that resistance curve.
And the cost of that isn't just financial, is it? What does that constant rotation do to the quality and, the safety of the crop?
It drastically raises the risk of chemical residue issues. I mean, farmers are under immense pressure to control these outbreaks, and the constant rotation, it's often pushed by just sheer desperation to save a crop.
So they might spray too close to harvest.
Exactly. It leads to situations where applying the next chemical they need might compromise the required safety harvest interval, or it could leave unacceptable residue levels on the produce. This is why the industry is so desperate for a zero residue, highly effective alternative.
And here's the crucial irony, right? Modern protective agriculture, the stuff that's supposed to be the gold standard for high-value crops, it actually makes the mite problem exponentially worse.
It creates a perfect storm. It really does. If we look specifically at the data on 32 mesh screenhouses, these are highly controlled environments.
Right, for premium crops like citrus.
Exactly. They're designed to exclude larger flying pests, like the Oriental fruit fly or the Asian citrus psyllid.
Yeah.
And they're great at that.
But the very structure that protects the fruit from the big insects.
It inadvertently welcomes the tiny mites.
Oh, wow.
Yes. The 32 mesh screens, by their very design, they significantly reduce air circulation. And this lack of airflow, combined with the often high humidity inside a screenhouse, creates this warm, still microclimate.
It's a paradise for mites.
For the citrus red mite, Panonychus citri, this environment is heaven. It removes all the natural environmental stresses they might face outdoors.
The numbers that detail this are just shocking. Tell us about the mite count inside those sheltered spaces compared to just out in the open air.
So the study measured the infestation during the peak period in October. The average count of citrus red mites inside the screenhouse reached an astronomical 578.9 mites per sticky track.
Per sticky track.
Over a 2-week period. And to put that into sharp relief, the average count found outside the screenhouse was dramatically lower.
It was a 96-fold difference.
Exactly. 96 times higher. This single data point is the clearest possible justification for the 20-year hunt for a biological solution.
Because in those environments, chemicals just don't work the same way.
They don't. Chemical sprays are difficult to apply evenly. The reduced circulation concentrates any vaporized residue, and that ideal environment just supercharges mite reproduction and resistance. The existing chemical toolbox is failing precisely where the agricultural investment is highest. You need something that's systemic, safe, and effective in these high-density, low-airflow environments.
I want to briefly pivot to the existing non-chemical alternatives because researchers have, of course, tried to find softer solutions. Our sources talk about using things like potassium fatty acid salts, or what we'd call potassium soap, especially for controlling mites on crops like papaya. What were the limitations there?
Well, the research showed that fatty acid salts, they do have some potential. Specifically, certain saturated carbon chains, the C10 to C12 and the C18 unsaturated chains, they showed pretty decent lab toxicity. We're talking mortality rates between about 61 and 87% in controlled settings.
So on paper, they look pretty promising as a quick knockdown agent.
They do, but the translation from the lab bench to the actual field is where the failure point really emerges. When you use commercial potassium soaps like a common coconut oil soap, in field trials, the control was only moderate. It was somewhere between 62 and 72% control, even after 2 back-to-back applications. And that level of control is it's not adequate for a persistent pest that reproduces so quickly.
And the most critical drawback, the real Achilles heel, was the eggs, right?
Absolutely. The mechanism of these soap-based pesticides, it typically relies on suffocating the active mites or disrupting their cell membranes. But the mite eggs are incredibly tough and resilient.
So the soap just washes off?
Essentially. The field tests showed that the soap solutions had no significant effect whatsoever on the mite eggs. The unhatched rate, so the number of eggs that failed to hatch after treatment, was below 14.4%.
So if 85% of the eggs survive the treatment, you haven't actually solved the problem at all. You've just delayed the next outbreak by a few days.
Exactly. You're guaranteeing the next generation will emerge almost immediately, and that forced the farmer into this extremely frequent, costly, and ultimately unsustainable cycle of reapplication.
It really highlights the fundamental limitation of so many of these contact-based treatments.
It does. The solution needed to be fundamentally different. It either needed to be one that could penetrate that egg structure or one that, when eaten, could just destroy the internal systems of the mite decisively, preventing it from surviving or reproducing. That realization is what drove this dedicated 2-decade search for a biopesticide with a unique systemic mode of action.
This incredible need for a game-changing solution brings us to the discovery itself. The search for a microbial agent, it often starts by screening thousands of soil samples, nature's vast chemical factory.
Right.
But the light rod bacterium, Photorhabdus luminescens, wasn't found in the soil. It was found in the gut of the Heterodictis nematode. What makes that location so uniquely challenging?
That's the most fascinating part of this research. This bacterium exists almost exclusively as a symbiotic partner within the digestive tract, or as the research calls it, the throat, of this one specific type of entomopathogenic nematode.
Okay, so just to visualize this, the nematode itself is microscopic. It's a tiny soil-dwelling worm whose job in nature is to hunt and kill larger insect pests.
That's right. So you have a microscopic worm that has to be found and isolated first. And then inside that worm is an even smaller, highly specialized bacterium that has to be extracted without any contamination.
It sounds less like a scientific search and more like a biological heist.
It absolutely elevates the technical difficulty. Yeah. For Dr. Shengfeng Jia and the team at ACTSRI, this process was just incredibly laborious. They spent a full year simply screening the nematode species Heterohabditis SPPAGI just to find one that was carrying a potentially useful symbiont.
1 full year just to identify the candidate worm?
A full year. That speaks volumes about the specialized knowledge required just to even begin this process from such a delicate host.
So what were the actual steps in the isolation? I mean, how do you reliably and cleanly extract a single bacterial strain from the gut of a creature you can barely see?
Well, this is where nature provided an incredible, really fortunate quirk that served as the researcher's map.
Okay.
The bacterium naturally emits cold light, bioluminescence.
It glows in the dark.
It glows in the dark.
Yeah.
The theory is this helps the nematode bacterium complex attract secondary scavengers in nature, or maybe suppress competitors. But for the scientists, it was a perfect optical signal.
So they didn't need a chemical stain or some elaborate piece of machinery?
No, they just needed the dark.
Wow.
Precisely. The entire isolation and the subsequent separation of the strain had to be conducted entirely in a dark room. The faint, steady glow of the Photorhabdus luminescens was the only reliable way for the team to visually track the presence of the bacterial colonies they wanted and separate them from, the thousands of other non-glowing microorganisms that would inevitably be present.
I'm just picturing that, working for hours, day after day, guided only by the subtle glowing signature. It requires just immense concentration and precision.
It really adds a human texture to the scientific process, doesn't it? It wasn't some mechanical discovery. It was a deeply manual, patient, and persistent effort, all guided by that faint light.
And that persistence led to a breakthrough.
A huge one. They didn't just find one glow. They eventually identified and successfully cultured at least 3 subspecies of the bacterium, and each one demonstrated different levels of insecticidal and miticidal activity.
Which then required a whole new round of testing.
Okay, exactly. Subsequent rigorous testing to optimize and select the most potent strain, which ultimately became the 0805 P2R strain that's now being commercialized.
When we talk about the commitment, we just have to stress the timeline again. The initial search for this nematode symbiont began around 2004. We're talking about a commercial launch in 2025. That's 2 decades. In an era where funding so often favors quick returns, what does a 20-year commitment tell you about the difficulty of biopesticide development?
It tells you that success in this field often requires an institutional dedication that just transcends typical market cycles. They weren't just looking for an organism. They were trying to decipher a highly complex, co-evolved biological system.
And then figure out how to mass produce it.
And then figure out how to stabilize and mass produce the active part of that system. Most quick attempts at biocontrol fail. They fail on stability or cost or regulatory hurdles. A 20-year investment indicates they repeatedly encountered and solved major engineering and biological problems, which shows an unwavering faith in the potential of this little glowing bacterium.
That kind of sustained effort is truly rare, and it's absolutely necessary for this level of fundamental innovation.
It really is.
So we've isolated the glowing assassin. Now let's get into the deep mechanics. How does a bacterium that thrives in a worm's gut kill a mite? What is the Photorhabdus strategy, and how has the commercial product harnessed that natural, lethal process?
Well, we have to understand the natural biological cycle first, this so-called Trojan horse strategy. It serves as a blueprint for the engineered product. In nature, the nematode is the delivery vehicle. It actively seeks out vulnerable insect larvae in the soil.
Okay, and how does the nematode get inside the host?
It invades the insect, typically entering through natural body openings like the spiracles, which are breathing pores, or the mouth. Once it's inside, it navigates to the hemicle, which is basically the insect's blood cavity.
And this is the moment the Trojan horse opens.
That's the moment. The bacterium is released.
Okay.
The photohabdus luminescens is released into the insect's internal environment. It then begins to multiply exponentially, just rapidly overwhelming the host's natural defenses. And crucially, within 24 to 48 hours, the bacterium starts to secrete its arsenal. Multiple potent, high molecular weight metabolic products.
And it's these proteins, not the bacterium itself, that deliver the fatal blow.
Exactly. They end up liquefying the host insect internally.
And the commercial breakthrough was realizing you don't need the fragile nematode, you just need to mass produce the chemical weapons the bacterium makes.
That is the core innovation. They bypassed the unstable delivery system and focused on the stable active ingredients. The bacteria produce what are known as toxin complexes, or TCs. And these TCs are the powerful metabolites that destroy the internal organization of the pest.
And for mites, the result is that catastrophic gut tissue rupture, the intestinally punctured and rotten effect we talked about.
Precisely.
Let's get highly specific about these toxin complexes. You mentioned they are exceptionally large proteins, bigger than 100 kilodaltons. What does that complexity mean for their lethality?
It means they're incredibly sophisticated weapons.
Yeah.
The TCs are not simple enzymes. They are massive protein machines. They're composed of 4 main components, usually labeled A, B, C, and D.
Okay.
Component A is often the toxic payload, the part that actually does the damage to the cell. Components B, C, and D form a complex structure that's responsible for recognizing, binding to, and delivering that payload across the host cell membrane.
So it's a highly specialized lock-and-key system designed for maximum efficiency against arthropod cells. How do we think the delivery actually works once a mite ingests the product?
So when the mite feeds on a treated leaf surface and ingests these dried protein metabolites, the toxins enter its midgut. The toxin complexes are specifically designed to recognize receptors on the cells that line the mite's intestinal wall.
And once they bind?
Once they bind, the complex uses its B, C, and D components. They often form a kind of injection system or a pore to breach the cell membrane and inject that toxic A component directly into the cell's cytoplasm.
It's an internal assassination. Instead of just disrupting the nervous system like so many chemical pesticides do, this literally causes the vital digestive tissues to collapse and rupture from the inside.
Exactly. And this is why the mechanism is so devastating, and it's also why resistance is theoretically much harder to develop.
Right, because a mite might evolve resistance to a single chemical.
But it's far more difficult to evolve immunity against a complex, multi-component protein machine that is actively destroying the physical integrity of its most vital internal organ.
We have data on the efficacy of these protein concentrates against major insect pests, not just mites. Let's look at the toxicity comparison using LC50 figures. For anyone listening who might not be familiar with technical toxicology, what exactly does LC50 mean?
Right. LC50 stands for lethal concentration 50%. It represents the concentration of a substance, in this case measured in parts per million, or ppm, that's required to kill 50% of the tested population under standard lab conditions.
So a lower LC50 number means it's more potent.
Exactly. A lower number signifies a higher level of potency. It gives us a great comparative benchmark for toxicity.
So how did the 100 kiloday protein concentrate from this bacterium stack up against well-known destructive pests, like the diamondback moth?
Against the highly destructive diamondback moth, Plutella xylostella, the tests showed really strong oral toxicity. It required an LC50 of just 56 ppm. That's a very potent result.
Okay, and what about another pest, like the greater wax moth?
Well, you can contrast that with the greater wax moth, Galleria melanella, which required a higher concentration of 200 ppm to achieve that same 50% kill rate.
That difference between 56 ppm and 200 ppm, that's a factor of almost 4 times. That tells us something really important about the specificity of these toxins, doesn't it?
It really does. It indicates that even though these toxin complexes are considered broad spectrum, their precise binding affinity to gut receptors varies significantly between different insect orders and species.
So the diamondback moth's gut is just way more vulnerable to this specific protein cocktail.
It seems to be, yes. And this kind of nuanced efficacy data is absolutely vital for deciding which crops the product is best suited for commercially.
And what about pests that didn't immediately die from ingestion? Did the toxins still provide some degree of control?
Yes, and this demonstrates a really important sublethal benefit. For pests like the corn earworm, even where the concentration wasn't high enough to be lethal, the toxins induced significant feeding inhibition.
So they just stopped eating?
They stopped eating. And a pest that doesn't immediately die but stops consuming your crop is a massive win for the farmer. It reduces yield loss during an active infestation. So the product can offer both curative and preventative protective effects.
The insecticide angle is clear, but the discovery gets even more valuable when we consider its dual role as an antimicrobial agent. I mean, why is a gut-dwelling bacterium secreting powerful antibiotics?
It's a matter of evolutionary competition. It's fascinating. Once the nematode releases the bacterium inside the insect host, the host dies, and that resulting corpse is a really rich nutrient source.
But it has to be protected from competitors.
Exactly. It must be protected from competing fungi and bacteria in the soil. So P. luminescens secretes this complex cocktail of metabolic products, chitinases, lipases, proteases, a wide array of antibiotics, specifically to suppress other microorganisms. It's essentially clearing out the house, so the nematode has a clean, protected cadaver to reproduce in.
And the researchers realized they could harness that protective mechanism for agricultural benefit outside of the insect corpse.
That's it. They tested this antifungal and antibacterial arsenal against common plant pathogens, and the results were hugely promising. The bacterium demonstrated significant inhibition against 9 out of 10 tested plant pathogenic fungi.
Give us some of the heavy hitters on that list. What kind of fungi are we talking about?
We're talking about serious commercial crop destroyers. Rose gray mold, Botrytis cinerea, which causes devastating rot, mango anthracnose, a major post-harvest disease, apple brown spot, pepper blight.
Wow.
Having a single application that kills mites and simultaneously combats these field fungi is a genuine value multiplier for farmers. It reduces the need for separate fungicide sprays.
And it even inhibits certain problematic bacteria.
Yes, it successfully inhibited 8 common and plant pathogenic bacteria. That includes even competitors like Bacillus thuringiensis, another commercial microbial insecticide, and pathogenic bacteria like Salmonella typhimurium.
So it confirms its unique position as a true multifunctional biopesticide.
It's tackling 3 classes of threats: acaricide, insecticide, and fungicide. This incredible complexity is what justified that 2-decade research investment.
We've established the potency and the complexity of the science, but science only becomes a solution when you can solve the logistical problems of commercialization. Our sources stress that the Taiwanese approach was innovative precisely because it solved the stability hurdles that had plagued other international attempts to commercialize P. luminescens.
This is maybe the most critical difference between a lab success and a market success. The previous international efforts, they tried to sell the entire natural system. The live nematodes carrying the bacteria.
Right.
But nematodes, as a commercial product, they have some really fundamental flaws.
Flaws that make them impractical for widespread farming.
Exactly. They are delicate living organisms. They require costly feed, often demanding high-grade sources like pig liver, which just dries up production costs. And more importantly, they have extremely low viability.
They die quickly.
Very quickly. Under field storage and transport conditions, they often die within about a month. Trying to distribute a product with a 1-month shelf life across an entire country, let alone globally, is a logistical nightmare. It just prevents competitive pricing.
So the game-changing innovation by Dr. Xi's team was realizing the delivery vehicle, the nematode, was the bottleneck. The true product was the content, those stable protein metabolites.
That's it. The innovation was separating the light rod bacterium from its host and then focusing on large-scale industrial fermentation to mass-produce the active, high molecular weight proteins.
Then these proteins are stable.
Biologically active, but chemically stable. The resulting product is a concentrated, stable formulation that can be stored at room temperature for up to 2 years.
That immediately turns a delicate, expensive logistical liability into a standard, shippable agricultural input. 2 years versus 1 month. That is a revolutionary change in the economics of biocontrol.
It democratizes the technology. It significantly reduces distribution costs, it eliminates the need for a complex cold chain for storage, and it makes the product accessible to farmers in remote regions who might not have access to specialized refrigeration.
Let's move from logistics to safety. You mentioned earlier that despite causing this internal rupture in mites, the product is completely safe for mammals. Why does this targeted mechanism, the toxin complexes, only harm arthropods and not us?
Well, the safety profile is exemplary precisely because the mechanism is so highly specific. Remember, the toxin complexes are these complex proteins designed to bind to and breach the midgut cells of the target pest. Mammals, humans, cats, dogs, we have fundamentally different digestive systems compared to mites and insects.
We don't have those specific receptors in our gut lining.
We don't. The lock-and-key system the bacteria evolved to target arthropods simply doesn't recognize or bind effectively to mammalian cells. And furthermore, even if small amounts were ingested, our digestive system, which is highly acidic, would quickly denature and break down the complex protein structure, rendering the toxin inactive long before it could reach any sensitive tissues.
And that's why the toxicology tests came back clean.
Exactly. They concluded the light rod bacterium and its metabolites are entirely harmless to humans and other mammals.
This safety factor also extends to the wider ecosystem, which is so crucial for modern agriculture.
It is. The product is certified safe for predatory mites, the spider mite's natural enemies, and other beneficial insects like bees. This means farmers can use this product without worrying about collateral damage to the natural control mechanisms they're trying to foster. It contributes fundamentally to integrated pest management principles.
What about the resistance problem that plagues chemical miticides? Since this mechanism is so powerful, won't the mites just evolve resistance to it eventually, maybe by changing those receptor sites?
Well, while no pest control method is truly impervious to resistance in the very long run, the complexity of the toxin complexes makes adaptation extremely challenging. Mites would need to simultaneously develop mutations that alter the specific receptor sites for all four components, A, B, C, and D in the complex, or develop some method to neutralize the entire mass of protein before it even binds. This is a much, much higher evolutionary hurdle than developing resistance to a single small chemical molecule.
And the field trials support that theory.
Yes, current field trials show no indication of rapid resistance development.
Okay, so let's ground this in field performance. Does this safe zero-residue solution actually deliver the knockout punch required to compete with conventional chemicals?
Yes, and that is why this is such a landmark announcement. Field results show the control efficacy against persistent pests like the papaya leaf mite is fully comparable to, and in some cases slightly better than, conventional chemical miticides.
What are the numbers?
Efficacy is consistently reaching over 70%, with the potential to get up to 80% or more under optimal application conditions. The safety doesn't come at the cost of performance.
And this leads us back to the crucial advantage for continuous harvesting crops, like papaya. Why is that zero residue benefit so transformative for these specific crops?
Just consider the logistical burden. If you are harvesting a fruit like papaya or strawberries every day or every few days, you can't apply a chemical that requires a 10 to 14-day safety harvest interval.
You'd be contaminating your own product.
Constantly. You'd be contaminating recently sprayed areas with fruit that's destined for market. Chemical use imposes these massive timing and logistical restrictions.
But with the light rod bacterium product?
Because it poses no toxicological threat, it can be applied right up to the day before harvest without any residue concerns. It completely eliminates that 10 to 14-day mandatory waiting period.
That's a huge economic advantage.
For crops that generate continuous revenue, this feature alone makes the product economically superior. It allows farmers to control an outbreak without having to sacrifice saleable yield.
We have some farmer feedback that really illustrates this practical impact. What did the collaborating papaya farmer say after she tested the product?
The farmer reported that in recent years, because of rising global temperatures, the severity of leaf mite infestations had escalated to the point where she was often forced to just abandon and uproot entire crops prematurely to try and get control.
Devastating financial losses.
Absolutely. But after integrating the light rod bacterium product, she provided a clear, straightforward testimonial. The control effect was very good. And that phrase, delivered by someone whose livelihood depended on this solution, it speaks volumes about its practical efficacy where everything else had failed.
The timeline for this product, the 0805P2R strain, is moving really quickly now.
It is. The official government announcement confirming its suitability for controlling papaya leaf mites was on January 3, 2025. It got its biopesticide license on February 6, 2025, and the commercial launch is scheduled for mid-March 2025.
That's a very rapid transition.
It shows the confidence that the regulators and the commercial partner, H&N, have in the product's safety and stability.
And what are the market ambitions? Are they targeting a niche organic market, or are they aiming for widespread adoption?
Widespread adoption. The commercial partner is targeting 20 to 25% of the entire Taiwanese acaricide market share. And crucially, they intend to price it competitively, very close to the cost of conventional chemical agents.
That's the key. That removes the historic barrier that so often prevents farmers from switching to safer biological alternatives that cost premium.
Exactly.
And what's the immediate plan beyond papaya?
The research team is already working on expanding its application scope to other high-value, high-infestation crops, specifically green onions and strawberries, which also suffer heavily from both mite and fungal issues.
Right, so they can leverage that triple action capability.
That's the long-term goal. To fully leverage its capabilities, acaricide, insecticide, and fungicide, to establish a domestic, multifunctional biopesticide cornerstone and secure all the necessary organic and friendly farming certifications along the way.
This success story of the glowing assassin, it isn't an isolated event, is it? It fits perfectly into this broader national strategy for agricultural sustainability. We're talking about a national policy goal of reducing chemical pesticide use by half over a 10-year period. This breakthrough just validates that entire policy direction.
Absolutely. The light rod bacterium is a high-profile example, but the principle behind it is integrated pest management, or IPM.
Right.
And IPM acknowledges that you cannot beat the evolutionary clock with chemistry alone. You have to combine precise monitoring, biological controls, physical barriers, and targeted limited chemical use only when absolutely necessary. And the data proves this integrated approach leads to superior outcomes while dramatically cutting reliance on harmful chemicals.
Let's look at some specific examples where IPM is already showing spectacular success, proving the strategy works even before this new biopesticide hits the market. Let's start with a foundational crop like rice.
The results from Taitung are exemplary. Researchers implemented IPM strategies in rice cultivation, combining rational seedling management, precise monitoring tools, and the use of non-pesticidal materials. And the results were just staggering.
How staggering?
They achieved a 74.7% reduction in the total active pesticide ingredients applied per hectare.
A 3/4 reduction in chemical load. That translates to significant environmental relief. But did this increase costs? That's always the fear when moving away from simple chemical sprays.
Quite the opposite. Management costs were reduced by 49.2%, nearly half. And here is the point that completely invalidates the old paradigm. The control they achieved for major rice diseases like rice blast and sheath blight was superior in the IPM plots compared to the conventional chemical methods.
Better results for half the cost?
For instance, rice blast incidence was recorded at 0.9% in the IPM area versus 1.2% in the control area. It's definitive proof. Less chemical, less cost, and better disease control. It's an operational trifecta that should drive rapid adoption once farmers trust the process.
It's all about building that trust through verifiable data.
It is. And the same principles are being applied to those continuous harvest cucurbits, like loofah and bitter gourd, where chemical residue is a constant challenge. Here, diversified IPM strategies, soil management like solar disinfection, microbial agents, sticky traps, lure traps, and highly limited chemical use achieved a 30% reduction in chemical active ingredient usage.
And a corresponding cost saving, I assume.
A 25% reduction in overall control costs. The shift is well underway, demonstrating that high efficacy and sustainability are not mutually exclusive goals. They're often achieved together through smarter management. The light rod bacterium just provides an incredibly powerful new tool for that management regime.
Beyond this new bacterium, what other biological control innovations are being leveraged in this fight against mites and other major pests? The use of natural predators is still a huge area of interest.
Oh, indeed. In mite control, using predatory mites is a standard biocontrol technique. And research often focuses on reevaluating local species. For example, local predatory mites in the Amblyseius largoensis group are widespread and act as generalist predators in many ecosystems.
But are they effective enough for a commercial release to wipe out an infestation?
Generally, no. While they're useful for maintaining a low background level of pest mites, they're less effective at decisive control during a heavy outbreak compared to dedicated specialist predators. The most notable one is the Chilean predatory mite, Phytocilus persimilis.
How does that specialist differentiate itself in terms of effectiveness?
Phytocilus persimilis is just a highly efficient, specialized hunter. It is capable of consuming up to 5 adult pest mites, or 20 eggs, every single day. The local species, by contrast, are generalists. They'll eat pollen and other small organisms. So the research concludes that while local species are vital for natural conservation and ecosystem balance, P. persimilis is the go-to choice for a commercial release that's intended for rapid, decisive control.
And finally, let's revisit the fungal control angle. We saw the bacterium has antifungal properties, but researchers are also using entomopathogenic fungi directly to control major insect pests, often specifically because they're safe for beneficial insects.
That's correct. Entomopathogenic fungi are a crucial alternative. Research is advancing on strains like CHF523, which target pests like the large, difficult-to-kill lychee stink bug. This fungal strain showed a high mortality rate, 77.6% over a 14-day period.
And why is fungal control so strategically important during specific periods of the crop cycle?
Fungi offer a vital microbial option during the critical flowering period. Stink bugs have to be controlled then, but applying broad-spectrum chemicals during flowering poses a lethal threat to essential pollinators like bees. A targeted microbial solution, like CHF523, allows farmers to control a major pest population without risking the bee populations necessary for fruit set. It just demonstrates this commitment to finding targeted, safe, and seasonally appropriate solutions that align with ecological demands.
This has been a truly comprehensive deep dive. To bring it all together, we began with the desperate problem of the chemical-resistant spider mite, an enemy that thrives, particularly in the modern, sheltered environments of high-value agriculture.
And the solution was unlocked through the 20-year commitment of Dr. Shi Fengji's team, who followed the faint glow of Photohabdus luminescens hidden in the throat of a tiny nematode.
The scientific and commercial breakthrough wasn't just finding the bacterium, it was the realization that the unstable, live nematode was unnecessary. By isolating and stabilizing the complex toxin complexes, those powerful protein metabolites, they created a product that delivers a decisive internal kill.
Causing gut rupture in pests while ensuring complete safety for humans, mammals, and beneficial insects.
This innovation solves that crucial logistical flaw of earlier approaches, allowing the product to be stored stably at room temperature for 2 years and applied without the typical 10 to 14-day safety interval. That provides an immense economic advantage for those continuous harvesting crops.
This impending launch in 2025 is a critical piece of the puzzle, aligning perfectly with the national strategy for chemical reduction. And that's a strategy already proven effective by significant reductions in chemical use and superior control results in rice and cucurbit IPM programs. The light rod bacterium is therefore a profound symbol of how decoding complex, co-evolved biological systems can yield targeted, safe, and effective replacements for broad, polluting chemical agents.
The idea that this singular biological agent offers a triple defense, as in a acaricide, an insecticide, and a fungicide, and that its success is based on extracting and stabilizing these large, multi-component protein toxins, not the living organism itself. It just suggests a massive shift in how we approach pest management. We're moving from crude chemical warfare to precision protein engineering.
The era of generalized chemical sprays is clearly giving way to highly sophisticated, biologically targeted solutions.
So here is our final provocative thought for you to chew on. Given that the light rod bacterium's success hinges on extracting these highly complex toxin complexes, these 4 component protein structures designed by nature and demonstrating their stability, what new opportunities does this specific breakthrough open for synthetic biology and genetic engineering? Could we, in the near future, use advanced techniques to design biopesticides from scratch, synthesizing stable protein toxins that perfectly mimic or even improve upon the complexity of these natural host-symbiont relationships, moving beyond the need for industrial fermentation of the original bacteria entirely?
That moves us from discovery into pure design. A fascinating potential future for sustainable agriculture.
That's all the time we have for this deep dive. We hope you feel significantly more well-informed about the future of pest control. We'll see you next time.